We are streaming, recording on
Hello,
live.
everyone. We are live again.
And wait, I will tell you maybe that works. Okay.
Did you find it?
Okay, attendees. Okay, I'm on it. I try to do everything at the same time. No, I don't find. I will just go on you.
I put it in the chat for you.
Oh, thanks. Perfect. Ah, sorry. Okay, Matthew, you can.
All right, we're all set. I'll give everyone a minute to get back and get settled. I hope everyone had a chance to refresh their beverage, to get something to eat, maybe to find a comfy spot, because we have some really exciting speakers come up in our next session. I'm really glad to see everyone coming back. Yeah, let's give everyone a couple minutes, maybe a minute, and we'll get started. Once again, this is Pint of Science 2020. This is a virtual Pint of Science Norway. Our first year doing this, so please forgive any issues that we have with technical problems. And as yesterday, we apologize again for not having live captions, but we will be putting transcripts on later for when we upload it to YouTube. So doing this virtually is obviously a bit different than having it in a pub, but I think I'm hoping everyone's having a great time. Now that we're back, we have Menon Bashar is going to talk about how digging up lakes today can tell us about the Vikings, and Agata Kershenska talking about life on Mars. So yeah, the format's going to be a little bit different than earlier today, since these are, as far as I know, we're not talking about Vikings on Mars. These are two separate talks. And so we'll have each speaker will talk for about 20 minutes, and then we'll have some time afterwards to ask questions. So please, anytime you think of them, raise your hand to ask a question, or type it in the Q&A and the questions part of the chat, and we will get to them later. So I'm going to stop talking, and I'm going to let Menon talk. She's a postdoc at University of Oslo, studying socio-environmental dynamics, soil climate, and human effects, and asking how looking at lake core samples can teach us about history. Menon?
Yes, I can share. Here we go, I think. So hi everybody, my name is Manon Bajar. I'm a researcher at the University of Oslo. I'm very happy to share this Pint of Science with you tonight, and I would like to thank the organization for this festival. So we are going to talk about the hidden face of lakes and how natural archive bring us to the Viking Age and beyond. So I'm working in a project that is called Vikings. So this project Vikings is about understanding how volcanic eruption impact the climate and how the climate impacted the Viking society, but more in general societies in the past. So considering the current climate change, it's very important to know how we can adapt our practices, our agricultural system to climate change. And to have some idea of how to adapt, we can learn from the past. So we look into the past, how was the climate, how was the society, and how the climate evolved in the past. And we look how the environment evolved as well, and how people reacted to this change in the past. So we learn from the past. So for that, we can look at archaeological evidence or historical documents, but we can also look at natural archives. Use these natural archives that can be tree rings, ice cores, lake sediment cores, and others, who will record information about climate changes in the past, also human activities, and how the environment evolved in general. So these archives are very useful. They are, for the lake sediment, continuous in time, while usually historical document or archaeology, they are punctual in time. But put together, they will complement each other. So today I'm talking more about lake sediment core. So this is the hidden face of the lake. So this part that we never see usually. So this is below the water. At the bottom of the lake, you have an accumulation of sediment. So this sediment, it's kind of mud, it's clays, it's sands, and these particles come from the surrounding of the lake. It comes with erosion, runoff, rivers. It corresponds to particles of soil.
I think we lost Manon.
To pollen, dust from the atmosphere. Okay. Is it okay?
Yeah, now it's better.
Yeah, I think you're okay now.
Okay. I hope it will stay stable.
In that case, so just sorry to you can in that case just turn off the webcam. That might help to just have the free of the presentation, not the video. Sorry for that.
Yes.
If it's not stable, it will help.
Okay, let me know. So I was saying that the sediment comes from the catchment. It can come from the pollen, dust from the atmosphere that are brought by the wind. That can be DNA from the vegetation and the animals around the lake. So this sediment, they accumulated year after year, and we can reconstruct by studying all this layer of sediment, how was the landscape around the lake year after year almost. So I will talk more about one specific lake that is located a little bit north of Oslo, a few kilometers south of the airport of Oslo. So it's called Lake Jokochean. It's a very small lake that is round. It has been formed 10,000 years before present, after the glacial retreat. So it's basically a piece of ice that has melted on the delta, the lowlands close to Gardermoen. And this lake is famous because on the shore of that lake, you can see on that picture that a big burial mound has been built here in the mid-6th century. And it corresponds to the largest burial mound of northern Norway. So this is an attractive place for archaeology and now tourism. So this lake has 18-meter depth of water, and below that we found the sediment. So this lake is round, and as it was a piece of ice that melted, there is out or in that lake. So it's only the rain and what is coming from the shore of the lake. So I will show you how we do the fieldwork and the lab work. I will show you how we date this archive and what type of analysis we did on that archive, and present you some results of that. So the fieldwork, it consists to take a sediment core, so it's like an ice core finally, but here we are going to fill a tube, a plastic tube with the sediment. So we just put the tube in the sediment. We can use a system with a hammer, and then it's like when you want to fill a straw with water, you just close the top, and then your sediment is staying in the tube, and you can bring it on the shore and then to the lab. We can do this from a boat or from a system, a platform system with two zodiac like this. and then it's quite easy to retrieve the sediment. We can also do it from the ice, but if it's too cold, it's a little bit difficult because it's frozen. Then we bring this tube full of sediment to the lab, and we can scan it. It's a CT scan like you can find in hospitals, and we can already see how it is inside the tube before we can open it. Then we can open the core with a system of saw. So then we will have two pieces, so two half core that we can describe, analyze, and sample for further analysis. So the sediment that is here a dark brown, it's very soft, it's wet, it's mud basically. And that mud can present this kind of lamination. Here we can see there's different layer, and this layer corresponds to time. So basically, one white layer and one black layer will correspond to 1 year, with a difference of color depending on the season. With, for example, the white layer for the accumulation of sediment during the spring and summer, and the black layer for the accumulation of sediment of the fall and the winter. And like this, we can go back in time as far as the lake exists. To date this sediment, we use radiocarbon dating, carbon-14 that is widely used. We can date the last 15,000 years with that method. So we date small piece of plant and wood that we can find in the sediment. We also use for the most recent sediment an isotope, the isotope of cesium, 137. So we can date the last century with that method. So in the sediment, in natural context, there is no cesium. This is an artificial isotope. So it should be, the activity should be 0. But in the recent sediment, we record 2 peaks in this isotope in the year 1963 and in the year 1986. So if we find these 2 peaks in the sediment, we know exactly which year it corresponds. So I will let you guess what can correspond these events, and you can answer in the chat if you have any idea of what it can correspond. What can be this pollution to the atmosphere that produced 2 peaks of cesium in these 2 years. A last method that we can use to date the sediment core is to use volcanic ash that we call tephra. It's these small particles of ash that are emitted when a volcanic eruption happens. The ash are transported with the wind and then will be deposited into the lake and then buried in the sediment. So if we know the year of the eruption and we find the corresponding particle in the lake, we can know that the layer is from the same year of the volcanic eruption. So then we can relate the depth in the sediment core to an age. So for Lake Yokocham, we have almost 6 meters of sediment. So 6 meters of sediment below the water. And you can see that these 6 meters of sediment, they cover the last 10,000 years. So at the beginning of the lake, 10,000 years before now, and then the sediment started to accumulate. So the sediment is not the same color and the same nature depending of the time, because the environment around the lake is changing. And if you look at this curve that represents the relation between the age and the depth in the sediment, it's not always constant. The slope will represent the relation. Here, the accumulation is faster in the beginning because there is no vegetation around the lake after the glacial retreat. Then there is more dense vegetation in the middle of the Holocene, so the period between 0 and 12,000 years before now. And then the slope is again very high because we started to have agriculture around the lake. So then there is no more vegetation, enough vegetation to stabilize the slope. So we have a more accumulation of sediment. I will focus on a specific period between 200 and 1300 of the Common Era. So it corresponds to the 3rd to 13th century of the historical period, because this is what we're interested in for the Vikings period and the Vikings project. So on that core, we analyze the geochemical composition, so different elements of the periodic table of the element with a scanner that will analyze with X-ray fluorescence this composition. And I will use calcium and titanium, two elements, to trace the change in temperature. We analyze also pollen that help us to reconstruct the vegetation and how the vegetation changed around the lake. And we also have indication of grazing activities because we can find also some spore of fungi that develop on the faces of herbivores. And if we can this, and if we find this ascospore husbandry. So here we have the record of this calcium and titanium that I used to reconstruct the temperature. So we are between 200 and 1300, between the late Iron Age and the Middle Age. We have the different period on top here. And we record this variation in calcium over titanium. So we measured it in a first core in blue and in a second core in orange here, because we wanted to be sure that what we record is valid for the whole lake and it's not just an artifact of measure. And then we compared that to temperature for the northern hemisphere globally, and it looks like it's vary in the same way. So here we have in Lake Lokojan a record locally of the temperature, a local record of the temperature. So then with that, I make an average and I transform it into temperature anomaly. So higher value are more warm temperature, lower value are colder temperature. And the zero is the average for the modern period, so nowadays. So between 200 and 1300, it was warmer between 200 and 300, and colder between 300 and 800. And then warmer again between
Manon,
800 and 1300. Warmer, colder.
Sorry to interrupt. Maybe you should turn off the video. We have a lot of quiz.
Okay.
Sorry for that.
Yes.
Okay, I can help with that.
Can you help?
Done. Yeah, done. Continue now.
Okay, super. So we have what we knew already before. We have a Roman period, the end of the Roman period that is warmer. Between 300 and 800, we describe it as a dark age cold period. And from the Viking Age, it's warmer and it's known as the medieval warm period. So, and we can see that after 800, the Viking Age starts with a climate warming. So it's possible that the Viking expanded at that time because the climate was warmer. Now, if we look at the vegetation change, we look at pollen. So this is percentage of pollen by category: trees, shrubs, poaceae, cultivated species, and herbs. And you can see that there is lots of trees between 200 and 800, and there is a major change in that diagram around 800. So we have less trees, so there is a deforestation, and more cultivated species. So there is a larger development of agriculture after 800 when the Viking Age started. And before that, there is also lots of yellow coming here from 400. So already an active period for agriculture between 400 and 700. If we look more into details in the different species, so in green here we have different species characterizing grazing activity, livestock farming, so urtica, noodles, rumex, and here this fungi from the faces of herbivores. And Below, we have cultivated species, hemp that was used to make textile ropes, and linum, rye, wheat, barley, and oats. So we can see that we have still a big chance at 800 with a lot of production of hemp and also cereals. And before that, we already had some pollens of barley, oats. But after that, it's less pollen, but it's then more indication of grazing activity. Then there is less grazing activity, but again, more pollen of cereals and hemp, and so on. Grazing activity, and then more pollen of cereals, and so on. So it looks like we have an alteration between cultivation of cereals and hemp and grazing, livestock farming. So I summarized that by this green box for husbandry, the period characterized by husbandry, and in orange for the period where it sees more pollen of cereals and hemp. And I did the same with the temperature reconstruction. I symbolized by blue boxes the cold period and red boxes the warm period, compared to the median of the whole period or only the period between 3 and 800. And what we can see is that when we have more grazing activities, more husbandry, it's also corresponding to the coldest period. Here you can see we have more cultivation of cereals, and it's where we have a little bit more warmer temperature, and so on. It's colder, we have grazing activity. It's warmer, we have cultivation of cereals. So it looks like there is an alternation between livestock farming when it is cold and cultivation when it's warmer. That can be explained very simply by when it's colder, you can't grow cereals. The season, especially in Norway, is too short to mature your cereals. So then you convert to more husbandry, more livestock farming. So this result suggests that there was an adaptation of the society to climate between 300 and 800, so during this dark age cold period. So we can then, like this, with that method, reconstruct the landscape evolution, the vegetation, and the climate for the whole period, for the whole last 10,000 years. Within the beginning, not a lot of vegetation, establishment of the vegetation, development of the agriculture with the climate. I wanted to show you another things that we find in that core. An important question for Norwegian is about fish. We are talking about lake, and Norwegian likes fishing, as my colleague Christer. And we had the chance to find at the complete bottom of that lake bones of fish. I didn't know it was bones at the beginning, and I had to ask an archaeozoologist who told me that it was bones of fish, sorry, and that was a freshwater fish, a roach in English, and it corresponds to the oldest freshwater fish found in Norway, dated 9,300 or 9,700. And I told you that there were no river coming in or out of the lake. So the question is still open to know how so old fish came into that lake. It's very rare to find that kind of fish bones in sediment core. It's only the second time. And it appears that the fish has been buried in the sediment. Here you can see that the layer is folded and has been buried by all this pack of sand in the lake. And from the origin of the fish, we have some hypotheses. It can be that it was so much fresh water melting from the ice, the glacier at that time, that the fish can come from South Denmark, for example, to Norway. Or it's also possible that a bird could have swallowed it into the lake passing above Norway. So that was it. I thank you for your attention, Skål. And I like to thank the Viking team I'm working with, and because it's a collaborative work and you need a lot of people to arrive to this result. Cool.
It's called.
That was a really great talk. I think it's really cool to learn about all these different ways that we can learn about history. You know, it's not just reading books, or it's not just looking at the artwork that we talked about earlier tonight. It's even things like looking at sediment and connecting those things. So I think it's really cool, and thank you for that talk and sharing it. Do we have any questions from the audience yet? I don't think so. Please.
Not yet.
Yeah, maybe you can mention how they can ask and interact with us.
No, if you have any questions.
Okay, what happened? We lost my.
Is it? Yeah, we don't have questions for now,
Yeah.
but maybe, okay. Yes, one. Adriana, I can open your.
So if you do have questions, you can type them, or you can.
We have one question. Hello. Yes, there's a. Okay, so the Adriana microphone should be open, so you can ask if you want. I don't know if she can.
Yeah, she
Hi.
did.
Okay. Hi.
Hi, Adriana.
Hi, everyone. So it was mentioned that for the laminations for the core samples, white corresponds to springtime and black for winter. I would like to know why. Like, how do we know that? Does that have to do with what falls in the lake or anything?
Yes,
Only
definitely.
For spring, for the vegetation, for example?
Yes, definitely. So it's exactly that. So for example, in spring and summer, first in spring, you will have the melting of the snow that will bring a lot of particles from the surrounding of the lake. So usually fine particles that are like clays and can be white. So it's a first thing that can be white into the sediment. It can be also the production of small particles in the lakes called diatoms, and that are also white. It can be precipitation of calcium that can be also white. And then in the end of summer, fall and winter, all the life in the lake will start to die, like the leaf on a tree. And then this organic matter will be dark and will accumulate. And especially during the winter, when the lake will be depleted in oxygen, a lot of organisms will die and will form this black layer as well. It's one of the examples that there is different process that can lead to this lamination. Sometimes it's precipitation of iron as well. Sometimes it's also the melt of glacier that will bring a lot of particles to the lake. So it depends on the functioning of the lake.
That's great. Thank you so much.
Thanks for your question. While we're waiting for some more, I had a question.
I do have a question.
Yeah, I know you had a question. Well, I think it's maybe both of us. We were talking, Dibble and I were talking about radiation in the chat. And I was wondering if, sort of looking forward, like if someone does this study in another 10,000 years, do you have any idea what they might find to indicate, you know, what's going on right now? Would that be in the record?
With radiation, you said?
Maybe radiation, or just is there something identifiable about what's going on now that would show up in this record? You know, if you know, your future self 10,000 years from now is doing the same study.
Yeah, I think so. We have still a lot to discover, and radiation, it's not so easy to find in the sediment. It will more influence the biology of the lake, and then will be seen in the sediment. I can answer the question in the chat about the radiation of Chernobyl, maybe.
Actually, yeah, that was my other question. You asked in one of your slides what happened in 1963 and 1986. So I was wondering, like 1986. Is that Chernobyl? We can detect at the bottom of the lake?
Yeah, definitely.
Okay.
Yeah.
Okay, nice. And
Yeah,
so
yeah.
what, I mean,
The fallout of Chernobyl, so it didn't stop at the border. It has spread all over Europe and and more than that, and it's visible in the environment. So it's very useful for us, I have to say. And so yes, we can see the peak in the activity of cesium still in the sediment. So in Norway, but in other lakes in France, it's also visible. It's visible, yeah, almost everywhere where the cloud has spread. So yes, in 1986 is Chernobyl, and we expected also to have a peak for Fukushima, but unfortunately, it didn't reach Europe enough to make a new nice chronomarker. And for 1963, yes, the answer has been given in the chat. So yeah, it corresponds to nuclear bomb tests. So this essay of atomic bomb, they started in the 50s and they ended in 1963. So just before it was forbidden to try this arms, this bomb, a lot of countries, they were like, okay, let's test before it's not allowed anymore. So there is a peak in 1963. So this happened in the Pacific and Polynesia, but it has been spread all over the world. So we have the same increase.
Great. Well, do we have some more questions from the audience? Some more raised hands?
No more questions, seems. No.
Okay. Well, cool. Well, thank you so much for this talk. It was really cool, especially being in Norway and getting to have that connection with the Vikings. And if you are around
thanks to you
in the area, you know, check out the Viking Museum and the History Museum and learn some more about that as well. Menon, if people are more interested in getting and learning more about your work, where should they go?
to learn more about the work. So
about
for the moment, we just have the website for the moment, and later when the project will be more advanced, we should have like an exhibition at the Viking Ship Museum with the new extension, hopefully.
Oh.
And yeah, hopefully more outreach when the publication will be out.
Great. Okay, we had one. Larissa, did you get that? Or Lavinia? We had a hand raised that I saw somewhere.
Yes, and this is Adriana. Now we can talk.
Hi. Hello. Sorry. I remembered I had a question. So I was thinking that if you had sampled any more lakes, and if all the information, if they all like agree, and if you like doing these studies, if you also like take information or look for information from other sources like historical writings or anything that might help you interpret your findings, or like vice versa.
Yes, so we are studying other lakes. A lake that is in Elverum, a little bit north. So that lake is now in study, so we don't have the results yet. But that lake is amazing because we can really count very precisely all the layer. It's called VARV in the sediment. So we will have a yearly resolved chronology. While the lake I show you, it's not layered all the way like this. So that's why I just have this radiocarbon age put together. So for this new lake, we will have a higher resolution. So it's the work of the PhD candidate Eirik Ballo that is also working with me. And in the Vikings project, we are also working with archaeologists working on the demographic in Norway at the same period. And so they will provide information about how many Vikings were there, for example, with climate modeler. So then we can compare the record of climate with the model, but of course not at the same resolution because we have local information for the lake. What model of climate will have like larger resolution. So South Norway, for example, or more large. We are also working with people working on ice cores. In Bern and in Oslo. I forgot. And yeah, and we are working also with tree rings. This is the work of Josh Bostich, that is working on old trees that were buried in the burial mound that I show on the shore of the lake. And with these tree rings, we can also reconstruct climate on the period of the tree, I can say. And on the sediment core, to confirm what I showed. And also on the sediment core I show, today we also have DNA analysis. So this is also a work in progress, and I don't have all the results yet. It's the work of a PhD candidate, Anna Terashur, and she's analyzing the DNA of the plants and the animals in the lake.
Perfect, thank you.
Okay.
Great. Thanks so much. Thanks, Manon. Manon shared her Twitter handle in the chat if you're interested in following that, and we'll check out your website. And I'm excited to see this work at the Viking Museum next time I go.
At some point.
Thank you. Thank you very much.
Thanks to you.
So from the bottom of a lake, we're going to go into the complete opposite direction and go to space. So Agata is going to talk to us about life on Mars. She's a postdoc at the Center for Earth Evaluation at UIO, studying what is needed for life on Mars and whether we need to go there or we can find out about this through meteorites. So Agata?
Hello, everyone. My name is Agata Krzesińska, and as it was said, I will talk about Mars, Martian rocks, and what we can essentially read from meteorites and rocks on the Earth without going to Mars or combined with going there. I think I'll stop video as my connection is slow. So I think that the question of are we alone in the universe was associated with humans from the very early our imagination, and naturally Mars, being the nearest planet to Earth, was one of the first objects for observation and to focus on. And already in 17th century, it was known that Mars has polar caps on the poles, which shrink and grow alternately associated with the seasons. It was observed that Mars has similar to Earth length of the day, also axial tilt, which is responsible for the seasons. So it was known that it is pretty similar to the Earth. And then in 19th century, in telescopic observations, it was seen clearly that on Mars, we can have some light and dark areas, which could correspond to oceans and continents. There were also speculations that it could be a vegetation because the color of the dark regions slightly changed in the seasons, which we now know is associated to dust movement. But at that time, people speculated that maybe it's some flora or plants influence. And also, some maps appeared which seemed to show that on Mars there are canals or channels which were speculated to be products of civilization. Now we know that it's just an optical illusion that these channels were put on the map, and actually they do not exist. But anyway, that was how the idea of life on Mars evolved. But actually, from the scientific point of view, expecting civilization as us is very very unscientific actually, because when we look at the Earth now, assuming and knowing, imagining that Earth is evolved planet with us intelligent life, when we look at Earth now, and when we compare biomass of the Earth, currently up to half of the carbon mass is contained in the mass of microbes such as bacteria. Plants and animals are negligible fraction only, and plants are also up to half of the biomass. And it's currently, but currently living species are also a little bit less than 0.5% of what was living on the Earth throughout time, and all other species are from out of over 5 billion living sometime on the Earth are now extinct. And do you think, could that be that species living before and now extinct were more evolved as what we have now? It's to answer that, I'll scale for you history of Earth to 1 year time. So the Earth formed some 4.56 billion years ago, and first water on Earth appeared 4.4 billion years ago. And if you are interested in absolute dates, they are listed here in the top of the slide. But I will scale it, and let's say that this 4.56 billion years ago was just New Year's Eve, midnight of 1st January, and what is now is 31st December, midnight. And all events, which we will talk about, are scaled up to this scale. And within this time scale, in mid-January, oceans appeared on the Earth, and it was kind of ocean planet till mid-February, and only in mid-February we have some record of oldest rocks. In beginning of March, in these rocks there are some indications which can suggest primitive single-cell life appeared, but these are very sparse evidences. And in mid-June, bacteria, so these simple microbial organisms, must have been common enough to change our atmosphere to produce oxygen to be abundant instead of carbon dioxide and nitrogen, which were before. And first eukaryotic cells appeared only in August in our time scale. So only then something other than microbes started to exist on the Earth. And if you heard about Aegeacaran biota, which is explosion of life with trilobites, which you might have heard, this in this time scale is only November. Dinosaurs were on the Earth only in 2 weeks in very end of December. Mammals appeared in mid-December, and we as humans are 15 minutes before midnight of 31st December. So actually, looking at that, Earth was always microbial life planet. And this means that even in our planet, life is just organisms which are barely seen by us. And this is why wherever we want to search for life, we have to look for these kind of organisms. But they are not easy to be tracked. And to find them, we may consider two concepts. One is biosignatures, so whatever they leave behind, like indications of existence or past presence of life. And also, we can understand what conditions in environment are required to support life, and this is called habitability. And an example biosignature is from the oldest terrestrial rocks. It's essentially geochemical evidence of presence of living forms. And in this aspect, when we look at carbon element, it has 3 isotopes, 12 with the mass 12, 13, and 14. And each geological process fractionates these isotopes based on mass. But biological processes, life essentially favors carbon 12. And in the 3.86 billion years old rocks, banded iron formation called, there were found some carbonaceous inclusions. The isotopic composition of each is increased in this carbon-12 as compared to other rocks, which can only be explained by the presence of living form at that time. So actually, the first evidence of life on Earth, biosignature evidence, is of that type. And for Mars, naturally, search for biosignatures was in heart of many missions. And we go to Mars since 1960s, and so far more than 20 orbiters and 6 in situ rover missions were there. We haven't returned sample so far. We might have it in next decades, but it makes our laboratory work difficult because whatever we want to do on Mars has to be done remotely. And search for biosignatures was done already by Viking missions, which was already mentioned that Vikings were on Mars, and it was in 70s years. And when Viking lander landed on the surface of Mars, it conducted 4 experiments designed specifically to look for biosignatures. And first experiment was to measure the molecular weight of chemicals which are present in this Martian soil. And surprisingly, the result did not show any significant organic carbon. Actually, the result showed that in Martian soil, there is even less organic carbon than it is in lunar soil, which was surprising. But in contrast to that, another experiment called labeled release showed some result that could indicate that metabolism happened in this soil. The experiment was essentially based on putting some nutrients into the soil, which were labeled with radioactive isotope, and then the track of this isotope was measured. So these 2 results were in big contrast to each other, which caused that essentially the mission did not provide any conclusive result. It was a lot of questions were left open. And only in 2008, by landing with another lander, some approach to maybe address this question or this controversy was found, that actually the soil of Mars contains a lot of perchlorides, which are highly oxidative, and any carbonaceous matter present in Martian soil may actually be destroyed. This shows that finding biosignatures is very difficult. And another approach to look for these kind of indications is to study meteorites. On the Earth, we have more than 200 meteorites, which certainly come from Mars. If you are interested how we know they are from Mars, I'm happy to answer that in the question and answer part. But meteorite Allan Hills 8401, which was found on Antarctica in year 84, is quite a unique sample in this term that it has very old age, which is different than all other Martian meteorites. And in this meteorite, there are some concretions of carbonates with, as you can see in this image, with zonal elemental composition of iron, magnesium, calcium, and manganese content in carbonate. And this kind of zonation in carbonates is supposed to be connected to biological activity. And additionally, in these concretions, we can find magnetite crystals, which are sometimes on Earth connected to some type of bacteria, which obviously this finding caused big interest in search for biosignatures in the meteorite. And one of such searchers showed this feature, which kind of resembles terrestrial bacteria, but it was of much smaller size. And later on, it was shown that this feature is actually only an artifact in the instrument, so it's not even real to the meteorite, while formation of these concretions can be explained entirely by abiogenic processes associated with thermal decomposition of carbonates and so on. Therefore, We can say that this search also provided no consensus about biosignatures, but it is supposed that this year 1996, when this finding was very hot, was the real beginning of exobiology as a science. And since then, we started to think about Mars as a planet which could really host life, or at least it was a topic which we should approach. But it's not easy to just find evidence of biosignatures and of life. So that currently we use another concept, which is concept of habitability, which means that in order to understand whether life can exist in any environment, we have to understand what this environment is and what kind of conditions are provided there, and if they can be conducive to origin of life. And essentially, whenever we have rocks in contact with water, it is likely that conditions were met for origin of life, and this information should be then retained in rocks. And life, I will call it as we know it, because we only limit our searches for life based on carbon chemistry and on water, has some limited actually list of requirements to survive. And obviously, life needs carbon for biomass to grow. It needs nutrients, but among nutrients, the most common are carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. And they comprise majority of required elements to sustain sustenance and for growth, while all other elements we can say are negligible. Obviously, life requires some energy sources, but in this aspect, life is very creative, and we can use, or as living forms, it can be used anything which is available, like sun, energy, heat, redox reactions, and all sources. Obviously, life needs space, which can be provided by porosity, cracks, or just surface. Needs water to deliver all the processes, and needs some dynamic to interact with the environment and some thermal regime. And all this information is retained in rocks if they were in contact with water and with living forms. So let's look at Mars today and try to find if it is habitable. Obviously, Mars is now dry with only small amount of water in the atmosphere. It's cold, so the temperatures on the surface do not allow water to keep liquid state. And additionally, it has very low pressure, atmospheric pressure, which is 1 of 200 that of Earth. Therefore, it's impossible to keep water on Mars. And also lack of or very thin atmosphere causes that the surface is bathed with UV radiation, which is destructive to life form, which means that Mars today is not habitable, is not hospitable to life. But Mars wasn't like that always. And if we look at the map of Mars, we can see something what we call dichotomy. On the southern part, this yellow on the map, called southern highlands, we can see a lot of retained impact craters, some of which are very big, while on the northern part, there are only few of them or much smaller amount of them. And in planetary geology, this fact is used to calibrate for age, because the idea behind that is that statistically, specific amount of impact events can happen in time. Therefore, craters form cumulatively on every surface, and old surfaces will be cratered with a high rate, with high amount of craters, while younger surfaces will be cratered or impacted by only few of them. And when it's calibrated with what is available to do that, I will not talk about details of the technique here. We can suppose that southern part of Mars is older than 3.7 billion years old, while the northern part is younger. And then when we look at the same part, but here with map, here with yellowish colors, we have southern highlands and northern part is blue. And all the dots show the detections of hydrated minerals. So the minerals which require water to form, like clays, carbonates, sulfates, and many others. And as you can see, they very much coincide with the oldest areas, which means that water was in contact with old rocks until 3.7 billion years ago on Mars, but not that much later on. And also in these ancient terrains, which we call Noachian, we can find geomorphological evidence that the water flowed on the surface. And these can be, for example, networks of valleys or rivers, or features resembling delta-like fans, meaning that water was flowing from the rivers to paleo lakes or sea-like structures, which all means that there was water on surface on Mars. So it means that Mars must have been warm enough to keep the temperature, and something changed at this point of 3.7 billion years ago. We don't know exactly what, but certainly before that period, Mars was warm and wet planet. And if we now come back to what I said in the beginning, that on Earth, first evidence for life is 4.0, 3.8 billion years ago, which on this scale is here. Then we realized that actually in time when life appeared on Earth, Mars was warm, wet, and probably habitable. So we may suppose that if only life could have formed on Mars at that time, maybe it still exists somewhere. But certainly at that time, there was chance for life to appear. So that now there is a couple of missions to Noachian terrains, which will study biosignatures, will look for biosignatures, and will try to understand the habitability of the environment. And one mission, which was launched in July this year, is Mars 2020 NASA mission with the rover Perseverance. And another one is ExoMars 2022, probably. So it will be launched in 2 years' time by ESA, European Space Agency. And the rover is called Rosalind Franklin. And both of these rovers will go to Noachian places. Perseverance will go to a place called Jezero Crater, while Rosalind Franklin will go to Oxyaplanum. And both these places are covered with vast deposits of clays and with some sediments suggesting presence of water, but these environments are different. And for example, when we look at Oxyaplanum, we see that the deposits of clays are quite homogeneous and there is some delta overlying it. While in Jezero Crater, it's a crater which was probably hosting a lake, and some delta finished in the lake, bringing sediments from elsewhere. Therefore, the context as for life may be different. And on top of that, as I said, both contain clay minerals, but these clays, from the spectral point of view, when we analyze them from orbiters, look different. And clays are actually quite complex group of minerals which form in a variety of different environment and histories. Therefore, it is important to understand what exactly spectrum says about the clay specific mineral and what's the then history behind that. And here you can have an example of two clay minerals, montmorillonite and saponite, which differ slightly in texture and in chemical composition of elements which build them. And in spectral signature, so from a spectrum which we could observe from orbit of Mars using the current orbiters, we can see that they differ slightly. But understanding of this spectra required some reference. And for that, we use analog rocks, which are terrestrial rocks for which we can perform a spectrum in laboratory on Earth and then compare it with what we know is a spectrum from specific place on Mars. If it matches, then we may suppose that the environments were similar. If it doesn't, probably it's a different type of mineral. And essentially, that's the goal of the project, which here in Norway, in our group, we conduct. The project is called Planetary Terrestrial Analog Library, and we build collection of physical terrestrial rocks that in some kind of mineralogy, so in kind of composition and minerals present, resemble those which we find on Mars or suppose we would find on Mars based on orbiter analysis. And having spectra of these rocks and putting them into library, we can support interpretation of the environmental conditions when we are on Mars and study Martian rocks. Here is the leaflet of our project. So if you are interested in how the library is built, you can visit the webpage or Twitter. But now we can come to conclusion. And I think the biggest take-home message which I would like you to remember is that life, as we know it, is not an evolved civilization of form, but it's microbes. And this is why science, from scientific point of view, we are looking for this life. It's not that we are modest or we expect Mars not being evolved. It's just life feature on planets. And Mars surface now is unhospitable, and we don't have any proof for life so far, but we know that when life appeared on Earth, the surface environment of Mars was different, much more habitable, and had liquid water and warm climate. Therefore, life might have formed on Mars, but presence of habitable conditions doesn't necessarily mean or imply formation of life. But habitability or the conditions can be read from the rocks if we are careful. And at the same time, search for biosignatures, which would be probably the conclusive answer, may be difficult because we need to understand environment again to understand if the biosignatures are destroyed or just different to read that we are used to. Thank you. I think I can stop sharing.
Thank you so much. Yeah. Oh, I just did something weird.
Okay. Okay, so now I'm back.
Thank you so much. That was really fascinating. And you did get to,
Yeah.
we did get to hear about Vikings on Mars. So,
Yes, they were.
so I like that. I don't see any questions yet. Remember, you can type your questions into the Q&A, or you can raise your hand to ask them. One question I had while we're waiting for some participants is, would we expect life on Mars to look different? And what I'm really asking is, if we were to like accidentally contaminate Mars, would we be able to tell if it came from Earth or if it was native to Mars?
It's not easy to contaminate Mars because there are very detailed protocols of that, and we cannot essentially everything which is sent from Earth to the Mars must fulfill some requirements that it's safe and not contaminating. And it's the opposite way around, obviously, as well. So that's the main problem with sample return mission, that anything which was in contact on Mars cannot be directly delivered to the Earth. So the sample, which will be taken from Mars and then captured by orbiter on Mars and sent to the Earth, the capture device cannot enter the atmosphere. Anything cannot enter the terrestrial atmosphere but the sample. And not that restrict, obviously, but similar protocols are for Mars. And for example, places which are supposed to host life at the moment, or potentially supposed to host it, are very restricted if we wanted to have a mission there. just in case to not mix life or to not contaminate. So technically, in science fiction, it is possible, but science and engineers, mission planners are aware of this issue and yeah, try to prevent from that.
Thanks. Do we have any questions from the audience? I know I see you out there. Or from the rest of our panel, do we have any questions?
Maybe a bit out of the topic, but when we study the history of life on Earth, on Mars, can we predict what will happen on Earth? See that no more atmosphere, no more water. So is that the future of Earth? Like in like 3 billion years, it will be the same? Can we answer that question already, or you can't?
I don't think I can answer that. It's not my specialty. It's more probably evolution style question. In terms of conditions on the Earth, it's also difficult to predict, right? Okay. Because there are some points in the history of Earth already in the past, which are kind of catastrophic, not evolution related. So these things we would never predict. And I don't think, if I understood your question correct, but I don't think we are able to properly predict or like model how life on Earth will evolve or how conditions on Earth will evolve. We can understand some changes which are related to cycles and or like step changes, but not really, obviously not everything, and it's too complex.
Okay, thanks. Maybe Manon, do you have a question, or is that related to the field or not at all?
No, I don't have question. But it was a great talk, Agatha.
Thank you.
Yeah, I really appreciate it. I mean, it's fascinating to think of, like, could there have been Mars, or could there have been life on Mars? And, if so, like, how would we find it? And this idea of like having it, checking in meteorites and checking on Mars and just doing all these things is of course really fascinating. And eventually if we ever send people there, of course, then we're going to have to be concerned with all these things even more, right? So yeah, so it'd be really interesting. I mean, it'd be amazing, obviously, if we found something indicating life. But either way, just like the amount of science that goes into getting to Mars and discovering things on Mars and analyzing all the things that we find on Mars is fascinating. So thank you for sharing this. If we don't, looks like we don't have any questions from the audience, and it's getting a little bit late. So thank you all for participating in Pint of Science. This is our second night. Tomorrow, I forgot what we were doing tomorrow. Do we have tomorrow's thing?
Okay, so tomorrow we go. So tomorrow we go from. Oops, I made it. Get review. Okay, what it will be good like that.
Oh, I have it.
So tomorrow
we have a talk about
climate change. We have a lot of topic.
So we have climate change talks tomorrow, and how we can solve it.
No, yeah, exactly. So tomorrow, like, will be like mainly focused on climate change. Will be at the different aspect of climate change. So it can be as well. We have an introduction at the start, but we will as well see more as well on the tree side, and as well how we are involved as a society. On maybe the last speaker may have some key opportunities of what we could do. So I'm looking forward, and I will share actually. Oh, I forgot to put that. Sorry, everybody. I will just put the link so you can directly see, or maybe Matthew already did it. I'm correct. Okay. I just copied the link where people can actually go and sign up for tomorrow. So thank you, everybody. I learned a
Yeah,
lot.
and if you want to learn more about what today's speakers are doing, on the sign up page, we have links to their websites and to their Twitter. And yeah, thank you again to our sponsors, to our hosts, to our speakers, of course, and we will see you all tomorrow.
Wait, we
No.
have actually in the queue, we have
A comment.
yeah.
What?
We have a comment, it's not a question, it's just a comment. So that, I don't know if Gabriela wants to talk directly. I can open your microphone. Maybe not, anyway.
Okay.
So the information is overwhelming and fascinating, a bit difficult to digest fast and ask questions. Great presentation.
Well, thank you for saying so. That's very nice.
Yeah, I can understand.
Yes.
Anyway.
All right.
We
Well,
are.
thank you all again, and hopefully we will see you tomorrow evening.